Developing a cancer vaccine has not been easy. There have been many failures, and I want to share with you how advances in our understanding of the immune system and new technologies may produce the first successful therapeutic cancer vaccine in a couple of years. Can the immune system even detect cancer cells? This was one of the first questions that needed to be answered, and now we know that it can. In fact, the immune system protects us from cancer cells all the time. Every time our cells divide, there is a chance for mutations to arise, and sometimes these mutations create cancer cells. And for the most part, the immune system can recognize and destroy individual cancer cells. You see, cells have a process where they take fragments of the proteins they have inside and put them on their surface to present them to the immune system. This is how the immune system examines the cell's contents. If the immune cells find something that doesn't fit the body's definition of what is acceptable, they will proceed to eliminate the cell. This could be a piece of the virus that infected the cell or a mutated protein. So we know that the immune system can detect and kill cancer cells by detecting mutations, but then why do we still get cancer? Just like viruses that figured out a way to evade our immune system, thriving cancers have also found probably more than one way to evade the immune system. For example, cancer cells can put the immune system to sleep by increasing the levels of checkpoint proteins, which are proteins that will signal the immune cells to stay inactive. This was huge when discovered, and drugs called checkpoint inhibitors that halt cancer cells from suppressing an immune response are now being used for treating some types of cancer. Another reason could be that the mutated part of the protein is not presented on the cell's surface. This way, immune cells have no way of telling if it is a cancer cell or not. There may be other reasons, and as they are uncovered, we can develop strategies to fight cancer cells more effectively. Even before finding checkpoint inhibitors, scientists tried to find targets to differentiate cancer cells from healthy cells. At the time, the best targets they could identify were overexpressed proteins in cancer cells. But the issue was that normal cells also expressed those same proteins, just at lower levels. So drugs targeting cells containing those proteins would also end up harming healthy cells that also had low levels of the same targeted protein. Identification of targets specific to cancer cells, such as mutated proteins, was finally made possible with advancements in genetic sequencing. As cancer cells multiply, they develop more genetic mutations, resulting in mutated proteins. These mutated proteins are known as neoantigens and are only present in cancer cells. Since only cancer cells possess neoantigens, they represent the closest thing we have to an ideal target. So now the goal is to develop treatments targeting cells carrying neoantigens so that healthy cells are not harmed. This is where the mRNA vaccines come into play. If a neoantigen is identified, scientists can design mRNA vaccines containing the sequences of these mutated proteins, inject them into the tumor, and boost the immune system response to identify and eliminate cancer cells. This is by no means an easy task. Scientists must identify which mutated proteins are the most likely to elicit an immune response. Were the immune cells not trained properly or being signaled to stay inactive? Are those mutated proteins even being presented on the surface of the cancer cells? If not, why is that? There can be many reasons, and identifying the cause for each patient and having the tools to do something about it is what will make a difference. And to deal with this complexity, scientists are looking to use artificial intelligence. For example, after identifying the neoantigens with genetic sequencing, AI tools can use existing databases to determine which of these neoantigens are more likely to not only be presented on the surface of the cell, but identify which will have a higher chance of inducing an immune response. Combining the advances in our knowledge of the immune system and its interaction with cancer, genetic sequencing, vaccine development, and AI, will allow a level of personalized medicine that we have never seen before. And this is so important, especially when treating cancer. All cancers are genetically different, and being able to design a treatment specific for the target found in one person is hopefully going to be the game changer we are waiting for. Is this going to be the cure for cancer? Probably not, but it is a very strong tool to treat and control cancers, especially aggressive cancers with hundreds or thousands of mutations that we can actually use in our favor to find a target. My hope is that in the next few decades we will reach a point where people will just need a few shots to keep cancer under control. If you want vitamin D and minimize the risk of skin cancer, check this video.